Today's topic is RNA interference, an approach towards improvement in agriculture through biotechnology. Now here we are going to discuss about this approach called RNA interference, which is meant for creating pest resistance in crops. We talk about a nematode, a parasitic nematode on plants called Melodogyn incognita, which infects roots of tobacco plants. And it is a severe threat for crop production because there is huge loss in agriculture because of infestation by this parasite. How is it parasitic in nature? It injects its salivary secretion in the plant cells and withdraws all the nutrients from the plant, making the plant diseased. The disease is called root knot disease. It forms such knot-like structures in the tobacco plant root, and this is leading to a great reduction in the yield. So there is an economic loss, huge loss. Effect on the plant is that the plant remains stunted, very poor growth, undergoes wilting, and becomes susceptible to many other diseases like fungal infection, etc. also can be seen. Now what is the approach to prevent this nematode infestation? The best approach found is RNA interference. I stands for interference. This RNA interference can be triggered by generating transgenic plants, by inducing genes in them, by incorporating genes in them, which would trigger RNA interference. We will discuss what it is in detail. So when this is triggered, there are RNAs formed which are capable of forming double-stranded structures which will prevent that mRNA of the nematode which is making it parasitic, which produces protein needed for the proper development of the nematode. So that would be silenced. That is the whole concept behind RNA interference. So it's a biological process. This RNA interference is found in all eukaryotic organisms as a method of cellular defense. This is one important point you have to remember. Cellular defense. We are not talking about natural immunity in the body. We are talking about cellular defense. So there is a strategy called RNA interference found in all eukaryotic organisms. When this approach was known, scientists got the idea to induce such RNA interference in organisms to prevent so many problems. So before we go to the detail of RNA interference, let me have a briefing on what is actually protein synthesis which keeps an organism functioning well. So the two steps are transcription and translation under protein synthesis. So whatever protein is synthesized in our body is a result of the gene that is on the DNA. So the gene is transcribed to an mRNA, and mRNA translates the instructions on the ribosome. Coming on the ribosome, it translates the instruction to protein. So this is what happens. From the nucleus, mRNA comes out, it comes into the ribosome. On the ribosome, it dictates the sequences of protein, that is the amino acids that comes up to make the peptide chains. So if this is happening normally, an organism is normal, but if there is any kind of failure in mRNA, the protein may not be formed. So RNA interference takes care of, the steps are here, you can read it. RNA interference takes care of cutting off this mRNA, silencing this mRNA, which is needed for the growth of the organism, because it makes protein needed for the growth. So if mRNA is disintegrated, proteins won't be formed. This is the concept we are going to apply here. We are going to silence the mRNA of a nematode so that the protein which it needs to survive in the host plant would not be formed and the nematode would die. So this is the basic concept. So how is in vitro processing done to generate that silencing process? So this technology for silencing a specific gene, which is a specific gene here, we are talking about silencing the gene of the nematode. So housekeeping genes, that means genes essential for the nematode to be parasite to the plant, and that is very vital for the development of the nematode. That gene is silenced. So we have to first get that gene which is responsible for the growth of the organism, of the nematode, allowing it to parasite the plant. So we have to silence that gene. So it is processed, isolated, processed and incorporated into plant cells. By tissue culture method, you generate such plants which have the strategy to silence the nematode gene whenever the nematode infects the plant. So the steps involved are, you isolate such nematodes from the roots of infected plants, maintain them, get mRNA extracted from the nematode. Which mRNA? That mRNA that is going to be very important for making protein needed for that growth of that nematode to allow parasitism on the host plant. So that mRNA is extracted. By reverse transcription, cDNA is made. Only the gene coding for that protein, that sequence we want. So the cDNA is synthesized, copy DNA or complementary DNA. Then that cDNA is made to replicate so that both the strands are obtained. Double-stranded DNA is obtained. Okay. Now this is that gene now, cDNA specific for that protein in nematode. So we are making both the copies of that DNA. DNA strand is double-stranded. DNA is double-stranded. So we make a double-stranded DNA by allowing replication. Now this is the most important step. This double-stranded DNA is altered in vitro. This process is done in vitro, not in an organism. It is done in laboratory. It is altered to prepare template. Template means the parental strand for synthesis of RNA. You know, transcription I already said. First step is transcription. DNA to RNA is transcription. So to make RNA, which should have two strands, we are using this cDNA in an altered manner. What is the alteration done? The one strand is as such and the other strand is inverted. Inverted. See, this is 3 to 5. So this is ligated here 5 to 3. So this is inverted and you make one gene. Using both the strands, you are making one gene. So this is a new gene constructed by inverting the original gene. This is one main step here. Now this one strand of that DNA, which is not one strand, is allowed to get incorporated into the plant cells which we want to alter. We are going to develop transgenic plant. So what are you using as a vector? You are using agrobacterium. We have done TI plasmids, nature of infecting plant cells. So we are using agrobacterium to allow the transfer of this DNA into the plant cell. How? This altered cDNA is introduced into plant cell by using TI plasmid of agrobacterium. You know, TI plasmid is tumor inducing plasmid. So we are disarming them. We are disarming the TI plasmid, taking away the tDNA area, the infectious area only of the tDNA, and incorporating our construct, this cDNA construct in that area. And then agrobacterium is made to infect the plant cell which we are culturing in laboratory. So this plant cell will generate into a whole new plant having this gene. Okay, so this is a very novel strategy. RNA interference is a very novel strategy to suppress any gene that you don't want to function, desirable gene. So this way it will prevent nematode infestation. How? This is going to act further. This double strand, this cDNA, inverted cDNA, that is the construct we made, that is going to trigger the whole mechanism of RNA interference. Even viruses can be used to manipulate. They can be manipulated and introduce this double-stranded RNA intermediates can be given into any host organism. Okay, so that kind can be done. We are here talking about infection by agrobacterium. Okay, now what is that fate of that DNA which has come into the plant cell? This is the plant cell, suppose. And what happens with this cDNA which was introduced by the tDNA? It will transcribe in the plant cell nucleus, and during transcription it will form mRNA. The mRNA will be such that it has got the areas of both the coding region and the template region. So you have RNA which has antisense RNA, which is called antisense RNA and sense RNA. Because these two strands are complementary to each other, the gene coding, because we have inverted it in this, So definitely the gene coding here is complementary to each other. So because antisense and sense strand are complementary to each other, they will form a loop here, a turn here, forming a hairpin like strand, hairpin shaped structure, because they complement each other. Now this will initiate RNA interference mechanism in the plant cell. How? In the plant cell cytoplasm, there is an enzyme called dicer. We don't have that much of detail in 12th, but we're just naming that enzyme here for your reference. Dicer enzyme will cut this double-stranded RNA into several pieces, and those small pieces, all duplex, double-stranded still, are called small interfering RNA, siRNA. This is the central to the RNA interference mechanism. This is going to work in the nematode body now. This is happening in the transgenic plant. Now suppose the nematode infests this plant and it starts feeding on this plant, genetically engineered plant having this gene. What will happen? It will absorb siRNA. The nematode will absorb siRNA. Now what happens in the nematode cell? You see here. So when it absorbs siRNA, the siRNA will enter a multiprotein complex in the cytoplasm of the nematode. There is a complex protein, multi-protein, having so many proteinaceous components called RISC. It is together called a RISC, R-I-S-C, the RNA induced silencing complex. So this siRNA will enter the RISC. Once it is incorporated into the RISC, the enzyme, there is an enzyme helicase present in RISC. It will cut the two strands. See how it is cutting the bonds. So it is called unwinding. The two strands unwind. Next, what will happen? This RISC will degrade one of the strands. Okay. So the sense strand will be degraded. You can see here it is getting disintegrated, degraded. So that will be excluded. And RISC will now only be with antisense RNA. So RISC along with antisense RNA will bind to the mRNA of the nematode. From where has this mRNA come? It has come from the nucleus of the nematode. It is the mRNA coding for the protein required for the nematode's growth in the plant cell and also its reproduction. So when this RISC along with the antisense RNA will bind to the mRNA, mRNA was healthy. So when it binds here, what will happen? There is ribonuclease enzyme also present in the RISC. It will cut the mRNA and mRNA will be fragmented. Can you see that? So now this mRNA cannot, dictate for protein. It cannot make protein needed for the growth of the nematode. So what will happen to the nematode? This whole thing is happening in the cytoplasm of the nematode once siRNA is absorbed by feeding. Okay, so what will happen? The mRNA is fragmented, nematode gene is silenced. This is called silencing. It will not work. That is why it will not translate. That is why it is called silencing. So nematode dies because that protein needed is not formed. So it dies. This strategy is now also applicable as a potentially new class of therapeutics for treating so many diseases in human, even in human. Diseases which are caused due to certain protein, those protein can be silenced by this nature. This is a simplified explanation for 12th class. So, you can see this mechanism happening inside the cytoplasm of the nematode. See mRNA coming out of the nuclear pore. This is the RISC. This is the siRNA being cleaved by helicase. One strand disintegrating. Okay, this is beautifully shown here. I got it done for a recap. You can see the person who has done this. So this is disintegrated. The other antisense strand will bind here to this mRNA. This is mRNA of nematode, which should have coded for the protein, but now it will not, because this is going to bind with it and it will disintegrate. The mRNA will disintegrate. So this is silenced. This mRNA is silenced. It is not going to make the protein. That's all about it. Very interesting topic. So I hope you understood. Thank you so much. Those who have not subscribed yet, I request you to subscribe and keep learning. Thank you so much. Take care.